All right, Chem 313, Lecture 16. We are starting to come down the home stretch at this point. The topic of today's lecture is going to be ATP synthesis. And if you hear the term oxidative phosphorylation, this is what it's referring to. So oxidative phosphorylation refers to ATP synthesis that is powered by the electron transport chain. So we've already been through the electron transport chain. Let's review it very briefly before we go on to how the energy of the electron transport chain actually drives ATP synthesis. As you'll see, it's quite simple. All right, let's review the electron transport chain. So we said before that we have our inner membrane of the mitochondrion. So if this is our inner membrane, we have 4 enzymes in the electron transport chain. They're referred to as respiratory complexes. And 3 of the 4 are also proton pumps. So here's what happens. We have complex 1. All right, I'm drawing this diagram so that the inner membrane space is at the top and the matrix of the mitochondrion is at the bottom. Complex 1 takes electrons from NADH and gives us NAD plus in return. And in the process of doing that, it takes this enzyme coenzyme Q and reduces it to QH2. All right, so complex 1, its reaction, in essence, is NADH plus oxidized coenzyme Q, react to give us NAD plus and QH2. I'm not keeping track of all the hydrogen ions here. There's also protons that are involved in the reaction. I'm not going to go over that. All right, so this reaction pumps 4 protons into the intermembrane space. And yes, the protons are actually hydronium H3O+, but it's easier to just write H+. Okay, so that's complex 1's reaction. The function of complex 1 is it takes electrons from NADH and puts them onto coenzyme Q. Coenzyme Q, the reason I'm drawing it in the lipid bilayer right here, is it is a lipid electron carrier. And it stays in the lipid bilayer, which is important because all 4 of our complexes are in the lipid bilayer. They're all integral membrane proteins. Complex 2. Complex 2 takes FADH2 and oxidizes it to give us FAD. And it also puts the electrons onto coenzyme Q, giving us QH2. So we can summarize the complex 2 reaction as FADH2 plus an oxidized coenzyme Q give us FAD plus another QH2. And we said before that complex 2 is the only complex that does not pump protons. Now, something you should note is that complexes 1 and 2 are independent of one another. It's not like 1 has to go first and then 2 goes. These 2 can happen at the same time. You can do either or. However, you have to run the reactions of complexes 1 and 2 before you can do complex 3. So what does complex 3 do? Complex 3 takes coenzyme Q and oxidizes it back to regular coenzyme Q, which gives us a pair of electrons. Where do those electrons go? They go onto another electron carrier called cytochrome C. So we said that cytochrome C is a protein electron carrier. And it's confined under normal conditions to the intermembrane space. And we said before that cytochrome C can only carry one electron at a time. So in order for our electron count to balance out, the reaction at complex 3 requires 2 cytochrome Cs. So the complex 3 reaction can be summarized as reduced coenzyme Q plus 2 oxidized cytochrome C react to give us plain oxidized coenzyme Q and 2 reduced cytochrome C. And we get 4 protons pumped. All right, lastly, we come to complex 4. So complex 4 is the end of the chain. So what does complex 4 do? It takes our cytochrome Cs, the reduced ones, and it reoxidizes them back to the oxidized form of cytochrome C. And that means it has to do something with the electrons. What it does with the electrons is it drops them off onto oxygen, forming water. So the reaction for complex 4 would be 2 cytochrome C reduced plus 1/2 O2 react to give us 2 cytochrome C oxidized plus H2O. And keep in mind that in order to make water, yes, you need hydrogen atoms. What ends up happening is it takes a couple of protons from solution. You don't have to worry about that. Complex 4 pumps 2 protons. So we can summarize the electron transport chain with the following flow of electrons. They start out at NADH or FADH2. They go on to coenzyme Q. These are the complex 1 and 2 reactions respectively. They go from coenzyme Q to cytochrome C. And then from cytochrome C to water. And water is stable. Water is the waste product of this entire reaction. And so what do we notice? We notice that we get 6 protons pumped into the intermembrane space for 1 FADH2, or 10 protons per NADH. Now you guys will think back a few lectures to when I said that FADH2 is worth approximately 1.5 or 2 ATPs when it's oxidized, and NADH is worth more like 3 ATPs. This is why. FADH2 gets you fewer protons pumped into your intermembrane space than NADH does. And so it's those protons that we pump into the intermembrane space that are actually going to provide the energy for ATP synthesis. All right, so we have completed the electron transport chain. Now we're done. What do we notice? Now we've got a high concentration of protons, which is equivalent to saying a low pH in our intermembrane space. All right, by low pH, I don't mean it's like concentrated acid, but I mean the pH is roughly 6, which is fairly low by biological standards. Meanwhile, in the matrix, we've got a low proton concentration and a higher pH. pH is around 7, so I don't mean like basic, but it's high compared to the intermembrane space. Okay? Well, why does this matter? It matters because of the Nernst equation that we've seen before, right? Whenever you have a difference in concentration of something across a membrane, you've got energy associated with it. In this case, the free energy of transport across the membrane we'll say from the intermembrane space side to the matrix side, is equal to what? RT natural log of proton concentration in the matrix over proton concentration in the intermembrane space plus ZF psi matrix minus psi IMS. All right, let's have a look at this equation right now. So we said that this is the free energy of moving from the intermembrane space to the matrix. So in other words, the opposite of the direction we've been pumping protons. We're talking about letting protons move this way, okay? If we were to allow a proton to move from the intermembrane space to the matrix, what would the free energy be? Well, we'll notice right here that the concentration of protons in the matrix is lower than the concentration in the intermembrane space. Therefore, this first term is going to be negative, less than 0. Okay, that's because whenever you take the natural log of a number that's less than 1, you end up with a negative number. Now let's take a look at the second term over here. The charge on our ion is +1. And what about our delta psi? So I haven't talked about the change in membrane potential across the bilayer, but notice how this diagram is drawn. We have all these plus signs on the intermembrane space side and all these minus signs on the matrix side. In fact, some textbooks even call the intermembrane space side the P side and the matrix side the N side. So where that terminology comes from is positive and negative. The matrix is at a very negative electrical potential compared to the intermembrane space. In other words, the matrix has an excess of negative ions in it compared to the intermembrane space. So this term is negative. And a negative times a positive means that the second term is also negative. So that means that the free energy of moving a proton from the intermembrane space back into the matrix is very negative. It's very favorable. So this means that you can power an unfavorable reaction by coupling it to proton transport from the intermembrane space back into the matrix. So in other words, you can think of the electron transport chain as just a series of pushing protons uphill, right? We get the energy from oxidizing our electron carriers and use that to pump the protons into the intermembrane space. Now we're going to let the protons flow back into the matrix and use that energy to make ATP. Okay, this slide shows you the diagram of ATP synthase. ATP synthase is the enzyme that is responsible for catalyzing ATP phosphorylation. So the reaction works like this. ADP plus a phosphate ion react to give us ATP plus water. And we said at the beginning of the metabolism section, the free energy of this reaction is ridiculously positive. It's like plus 50 kilojoules per mole. So what we're going to do is make this reaction favorable by coupling it to movement of 3 protons from the intermembrane space back into the matrix. So if you wanted to, you could write the reaction as ADP plus phosphate plus 3 protons in the intermembrane space react to give us ATP plus water plus 3 protons in the matrix. And the overall delta G, it's right around 0. It's actually slightly negative, so this reaction does happen spontaneously, okay? So it's, I'll say, slightly negative. and therefore ATP synthesis is possible. All right, so this is the ATP synthase molecule. I have decided not to lecture at great length about how it works. I instead refer you to a nice animation. I'll post a link on Canvas that shows how this enzyme works. It's a really fascinating molecule. We've got two different parts to it. We've got what's called a stator, which is this transmembrane segment shown in purple, this long red stalk, and this collection of, I believe it's 6 subunits that sit inside the intermembrane space, or sorry, in the matrix side. So we have the intermembrane space at the top and the matrix at the bottom. So the stator, which is all of this stuff, it's fixed in space. It doesn't move. Then we have another part called the rotor, and attached to that is the rotor shaft. And so this is a complex of different proteins. The rotor actually rotates. And so what causes the rotor to rotate is binding of protons at certain exposed amino acid side chains. So it simply protonates certain side chains. And that creates a repulsion that causes this whole thing to rotate. And then after it rotates one full revolution, the protons come out at the bottom. And so while this whole rotor shaft is rotating, what it's doing is it's squishing together ADP and phosphate inside the stator part down at the bottom, and we squeeze out ATP as a product. So I don't care that you know all the ins and outs of how this protein works. It's a fascinating molecule. But what I care that you understand more is what is our reaction that is taking place here and why is it favorable or even possible? Okay, and as for how ATP synthase works, just have a look at the animation and I think you'll be wowed. Okay, I include this slide because you guys have a homework question related to it, and that's the topic of respiratory uncouplers. So what are uncouplers? Uncouplers are these molecules that transport protons back into the matrix by bypassing ATP synthase. Essentially what these are is lipid-soluble weak bases. Or I should say weak acid and bases. So what ends up happening is the uncoupler floats up to the intermembrane space side of the membrane and picks up a proton to become a protonated uncoupler, where it then diffuses back to the intermembrane space side and gives up its proton. And it can do this because it has a pKa that is near neutral. So that if it touches the intermembrane space side where the pH is slightly low, it will become protonated. But then when it drifts back over to the matrix side where the pH is above its pKa, it'll be deprotonated. So what ends up happening with uncouplers is that the protons can go from the intermembrane space to the matrix without passing through ATP synthase. And so what ends up happening is we use the electron transport chain to push the protons into the intermembrane space, and then they simply just leak right back where they came from, and we don't use that energy for anything. So what do you think is the effect of this process on ATP synthesis? And what do you suppose happens to the energy of moving a proton from the intermembrane space back into the matrix? I'm not going to give you the answers to those questions, but that's something to think about for the homework. In the normal situation, if you don't have uncouplers, the energy is used to make ATP. If you don't have ATP synthase, if you're just letting it leak across the membrane, the energy has to go somewhere. Where do you suppose it's going to go? All right, we've finally synthesized ATP. All right, we are done making energy through aerobic respiration. We've completed the whole cycle. So let's do some accounting. Let's figure out how many ATP per fuel molecule we get. All right, let's say we're going to use glucose as our fuel. So in glycolysis, you'll recall that for one glucose, we get 2 pyruvate, 2 NADH, and 2 ATP. In pyruvate dehydrogenase, we take our 2 pyruvates, get 2 NADH, and 2 acetyl-CoA. Okay, plus 2 CO2, but we're not going to be keeping track of that on this slide. And then those acetyl-CoAs go into the citric acid cycle, and we get 6 NADH and 2 FADH2 total, and 2 ATPs total. All right, so that means that for breaking down one molecule of glucose all the way to CO2, we end up with 10 NADH. We have 10 protons pumped for every NADH. We get 1 ATP for every 3 protons that leak back across the inner membrane. So we get roughly 33 ATPs from all of our NADH that we made from glucose. If you do the same math for FADH2, you get 4 ATPs from FADH2. Okay, so that's 37, but then we also have 4 ATPs that we just get as a bonus. So overall, we get 41 ATP per glucose total. That's the best case scenario. As we're going to see in a second here, you never actually get that much. All right, that's glucose. Let's do a fatty acid. So let's do palmitic acid, saturated fatty acid, which makes the math really easy. All right, so from a 16-carbon saturated fatty acid, in beta oxidation, we get 8 acetyl-CoA, 7 NADH, and 7 FADH2. All right, great. Then we take those acetyl-CoAs, and they go into the citric acid cycle. And 8 acetyl-CoA will end up giving us 24 NADH, 8 FADH2s, and 8 ATPs. So in total, we have 31 NADH times 10 protons per NADH times 1 ATP for every 3 protons gives us 103 ATP from NADH. Do the same thing for FADH2, you get 30 from that. And then you've got your additional 8 just as the bonus from the citric acid cycle. So we have 141 ATPs per fatty acid total. Again, this is the best case scenario. In reality, it's never that perfect. All right, so what do we notice here? We notice that a fatty acid gives you a lot more ATP than glucose does. And that's not really surprising because remember, we have 6 carbons to oxidize up here in glycolysis and 16 carbons at the bottom. However, if you were to take the ratio of ATP to carbon atoms, you would find that the fatty acids still give you more ATP than the glucose does. And why is that? Well, there was a homework question related to this. The reason for that is that the fatty acid is very reduced. It has lots of carbon-hydrogen bonds. The more reduced it is, the more we can oxidize it, the more NADH and FADH2 we get, and the more ATP that we get out of that. That's why fat has so many more calories than sugar, because it's more reduced. All right, now keep in mind this is a best case scenario once again. The actual ATP yield is lower. The general rule of thumb is that under normal conditions you'll get about 32 ATP per glucose. And the reason for that is because there's inefficiencies. Some of our enzymes are not 100% efficient, so especially ATP synthase. You also have to transport ATP out of the mitochondria and ADP into the mitochondria, and that ends up costing you a little bit of energy. You have to pump pyruvate into the mitochondria so it can get to pyruvate dehydrogenase. That costs you energy. So we have all these other processes that we've skipped over in this course, and those end up taking up some of our ATP. Nonetheless, the point remains that fatty acids are a better ATP source than glucose. Okay? That doesn't mean that fatty acids are better under every situation, because fatty acid metabolism has its own quirks. All right? We said one such quirk is that you cannot break down fatty acids as energy in the absence of oxygen. Okay, so there's all of our ATP that we've been able to synthesize. So what we're going to talk about in the next lecture is one metabolic use of ATP. And that is glucose synthesis. So that is our only example of an anabolic pathway that we will look at in this class, and that is chapter 17. And the official term for glucose synthesis is gluconeogenesis. So that will be the topic of our next lecture, and that way we will have covered both anabolic and catabolic pathways. So in the meantime, I encourage you to review the material on gluconeogenesis and take a look at the new homework assignment. And we're going to finish up metabolism on our next lecture.